Let's get started. Good evening, everyone. Hope you had a great day today. So today we are going to have a bit of practice with real data for visualizations and plotting ERPs, and we will get an intuitive picture of what is meant by time frequency representation of a signal. And Yesterday's lecture we were discussing few pre-processing. Last lecture we were discussing few pre-processing steps. So lot of terms might seem very complex, but today we will go through each one of them and get a deeper intuitive picture of it. I'm not going to show the equations or anything, and if time permits, we will discuss some of the noise sources. So hope everyone has downloaded or checked the notebook that I have shared in the last class, last email, sorry. Can I see some hands, whoever has tried downloading or tried executing the notebook? Okay, cool. I see two, three. Okay, anyway, so it's not a problem even if you haven't seen that. We'll go through line by line, and after the class, you can try it yourself. So that's not a problem. So let's start with a bit of recap from the last class. So in the last class we have seen what are the sources of EEG and what kind of information these electrodes on the scalp collect. So we have gone through a dipole analogy and the current sources and sink in the cortex due to the pyramidal neurons close to the surface, and their synchronous activity cause act like a dipole, and essentially we are measuring the electric field or potential due to that using these electrodes on the scalp. And then we have seen a big picture of how an EEG acquisition system looks like, and we have gone through a general data structure of continuous EEG data, how it is segmented into epochs, and we will be doing this in today's practice book also. And we have seen it is usually a three-dimensional data structure with channels, time samples, and trials on three different axes. Then we have seen two of the fundamental measures that we get through EEG, that is event related potentials and oscillations. One second. Okay, you can lower your hand, whoever raised. Okay. So whoever joined right now, we are just going through the recap of the last lecture, where we have seen the sources of EEG. One second, I'll take my pointer. Yeah. EEG acquisition system, a general data structure of how EEG is stored or represented in terms of channels, time samples, and trials. Okay. Yeah. And we have seen ERP characteristics and the oscillations. These are the two fundamental and direct measures that we get from EEG. Of course, we have to do a few processing steps, and we have also checked a few of them in this and what they mean, but it's a broad view, and there are a lot of terms, and in today's class we will clear that each one. And before that, we will see how a continuous EEG data looks, how do we visualize data, and how can we see ERPs using a real-time data. Okay, so before getting into the notebook, let's see what the data set is about. So the data set that we are going to use today is based on an audio visual task, where in the first few milliseconds, the participant is presented with a checkerboard on one side of the visual field in the sense on the computer screen here, either on left or right. And then after a certain delay, they are presented with the auditory stimulus, either coming from left channel or the right channel. And on few trials, they are presented with the smiley face, and whenever they detect a smiley face on those trials, they have to press a button. So this smiley face occurs rarely on few trials. So this is a task, and this is a sample task the participants were performing, and the EEG data was recorded. And now we are going to use the same data for our practice. So one second. Okay. So yeah, so I was whoever joined right now. Uh, so I have just recapped whatever we covered in the last lecture, and this shows what all we covered in the last lecture: the data structure, uh, the sources of EEG, and what are the direct measurements, and some of the pre-processing steps. Today we are going to practice with a real EEG data using a notebook that was shared in the last email, and then go through some of the details of this pre-processing steps so that uh, you're more familiar with what is exactly happening, and the terms don't uh, look too complex to you once we go through that. So any doubts in this task design?
Deepak, could you remind what was the purpose of the task? As in, what are we trying to find here through this
Okay, this is just a sample
task
task
design?
to see auditory evoked potentials and visual evoked potentials. So for that, on each trial, so like this structure of trials are shown to subject. So on each trial, what happens is for a few seconds, uh, a checkerboard type of stimulus is shown for a few milliseconds, and then there is a small delay. After a small delay, an auditory stimulus is shown to the participant, and these stimuli occur either on the left side or the right side. And there is no per se a scientific purpose for this task. It is just to get a toy data set of EEG and try to check the analysis. Okay, yeah. And whenever a smiley comes, the subject has to respond on this window. As soon as it disappears, they have to respond. And this smiley face occurs very rare in the trial, like in a after a few trials randomly. Okay, so now we will get into uh, the practice of with this with a notebook. So I hope most of you have at least downloaded the uh, notebook. So I will just go through hands on. If you have it open on your systems, you can also go through along with me. If you don't have it open, it's okay. You can do it after the class also. Okay. So I'll just change my screen.
Deepak, can you make me the host?
Yeah, yeah. Sorry. One second. Where is it? Yeah. Yeah. Okay. So okay. Oh, I think I have to share the entire screen. Okay, fine. Okay. Is my screen visible? Yeah. Okay. So, uh, is the notebook visible to everyone? Is the font size fine?
Yes.
Okay. Great. Okay. Sure. So, as I discussed, we have seen the task now. So, everyone has to, like, it might take some time to do the first few things. So uh, first one, basically we are going to use a library called MNE library, which is mostly used for neuroimaging analysis or visualizations in Python. So what we are doing here is just installing the MNE library to the Google Colab notebook. So you have to first open a Google Colab notebook and open the file that I shared with you, the IPython notebook, and install this library. And some of these are importing other libraries which are pre-installed as a part of Colab. So this one, installation of these four libraries, that is Matplotlib, NumPy and Pandas, you don't have to worry about. only thing you have to install is MNE library here using this command. Okay, I hope it's clear. So library, if who is not familiar, they're just a set of packages and programs which are written based on Python, so that you can directly use their functions using a object oriented calling. So we'll get it clear as we go on, but you don't have to worry right now. Just have to run these two codes, and then here, okay, and this data set is a combination of EEG and MEG data set, and we are going to use only the EEG part of it. Okay, so what we do is I have preloaded it right just before the class. So here it is taking the file and downloading it from the OSF web page where the data is stored right now. And then we also download an events file. So this is an events file path. Okay, sorry. Okay, fine. Yeah. So in this line, this is important, where this command mne io is nothing but mne has a internal package called io, that is input output package, which can read a file. So here the raw file is nothing but the EEG MEG file. So you give the raw file here, and using read raw fif. So this file is a FIF file, FIF file. For now, you don't have to worry what are different formats, but it's just an extension like a PDF or PNG. And then you also read the events file. Events are nothing but the triggers that we were discussing yesterday. So as the task goes on, there are different events. Like we have seen in the task, there is a stimulus, there is auditory stimulus, visual stimulus, then a smiley appears and a response appears. So these are all termed as events. So these are also stored in another file named this way, sample audio vis filt, this one. So basically in this cell, you are loading the events and EEG data, and just for the just for practice, we are cropping the signals to only 90 seconds. 90 seconds, that is 1 minute 30 seconds. And we have events also. We are reading the events based on this cropping, because we don't want events, we are not interested in the events beyond 90 seconds. Okay, so hope this cell is clear. Any doubts here? Until now we are just loading the data. Okay, so any inputs or anyone facing any issues with the notebook, please let me know if you have right now.
So we are importing the data, or sort of cutting the data between 0 and 90 seconds.
Correct, yes, exactly.
Is it so?
Yeah, correct, yeah. So this is just loading and reading the data, and then when you run this, so basically, as I mentioned, this data is a combination of EEG and MEG data set. So from the raw, which is the data, we are picking the EEG part of it, and raw.info will print the information of that data. So when you run this cell, yeah, it shows different information regarding that. Participants and experimenter are unknown because we want to keep it anonymous. Sampling frequency is 150.15 hertz. These are the channel information. It's a 60 channel EEG, and one of the channel is found to be bad. So right now what we're dealing with is already a pre-processed data. It's not, we're not doing any pre-processing right now. So for now you take it that it is already pre-processed, and now we are just going to visualize and plot the ERPs. And as we discussed yesterday, high pass, low pass, which will be clear right now. Yeah, so Atharva mentioned some doubt is there? Okay, fine. Okay.
Uh, Hello.
Yeah, Atharva.
Yes. No. Somebody asked this FI. What is FIF extension? So I just posted a Stack Overflow link.
Okay, okay, cool, cool.
Other doubt is what is the name of dataset we are using?
Oh, name of data set. So it's a data set which was available in sample data sets of MNE library. So it's called MNE sample data processed. So this is a file that this command downloads from this website where they have uploaded the data. Okay. It's an audio visual task which I have explained previously. If you have missed, we'll come back to it. So we'll just go through it and we'll try to visualize the things and then we'll go get there. So briefly, the task is they are presented with two kinds of stimuli. One is visual stimuli and one is auditory stimuli. Okay. So that just that information is enough, and it's a toy data set. It has it has no hypothesis to solve using EEG or anything else. Okay. So here we are just visualizing the sensors. So from the raw data which is stored in this object raw, we are plotting the sensors. Okay. So these functions are already pre-present in the library, MNE library, and MNE library is something which is being used for our purpose, and it is mostly used for neuroimaging data, any kind of neuroimaging data, EEG, MEG, MRI, even local field potentials, spike rates, etc. Okay. So now when you run this cell, you get their channel location. So as we discussed, there are 60 channels. So this is a head, and it's facing, the nose is represented by the sharp triangle, and these are the ears. And it shows the 2D, in a two-dimensional plane, the location of different electrodes on the scalp. So these are the electrodes, and as we saw here, there is one bad electrode which is marked in red here. Okay. I hope until now it's clear. There's nothing much with it. We just loaded the data, got the information of the data, and visualizing the channels. Okay, fine. There is some doubt. I just give me a minute.
So
Can we see
RK
some
Goyal
sample
asked
records?
that can we see some sample records in the data set? That's what you are showing right now. So probably explaining the data set might help, like why, how this FIF file stores things and what kind of data sets may go into it. That will
Oh,
give them an idea, like so this is a this is a data structure which we can't read through other softwares. Like you need this MNE uh, plugin to do that, right?
yes, yes.
Any library is
So there are many file
so.
formats. Is the question about file formats? Then there are like European data formats. We might discuss later, but I haven't made any material for that. So if time permits, I can look into and we can go through it. But for now, like it's just a FIF factorial image file format. So it's just a zipped version of some data. So yeah. Okay.
And you will show that
Yeah.
the descriptive statistics of some of the fields or some of the fields in the next few minutes. Another doubt is that we can change the channel names to standard channel names, right?
Okay. So usually there are many companies which manufacture this EEG caps uh, In the early days, it was only 1020 system, but later when high density EEG and different formats came and different companies are coming up, so they have their own system. So there is always a file associated with their locations. So EEG, their location file has their electrode locations and mapping with like which electrode is closest to the 1020 system, so that they can have which region is giving this type of signal, or some atlas where they can register or co-register the signals. Okay. Oh, cool. So here what we are doing is re-referencing. I will let you know what is re-referencing doing and why we have to do. For now, like you can, someone asked how does the data look. So here it's a time and we are plotting for 5 channels. Only 5 channels we are taking for now, just for visualization. And you see like this is a 1 minute 30 second of time. So you have this data, although we are cropping it, we just took the whole data here. So this is how the 5 channel EEG looks like. They look almost similar because they're all close by neighboring channels. So as we have seen yesterday, there is volume conduction and similar things come up close by. So okay, what's a channel? It's nothing but an electrode, and each electrode is interchangeably used as channel. So you can either say it's an electrode or it's a channel. Okay, yeah, cool. So yeah, so after re-referencing, you will eliminate some of the noise, and this is how the actual signal looks like from the 5 channels of interest. Okay, there is not much of a difference, but as we discuss the re-referencing in the next few slides, we will be more clear of what is exactly happening here. But for now, this is the data set for 10 seconds plotted here. Someone asked some function thing. Get data. Okay, we'll go through it to the end. Okay, hope this is clear. Right now we are just visualizing data for 5 channels and what is the effect of re-referencing. We will understand what is re-referencing doing. So what is re-referencing is nothing but for now in a brief, it's like you take average of all the electrodes, and for every time point you'll have one value for that average of all the electrodes, and that value you subtract from each of the electrode, so that a common sources of noise are subtracted in that electrode space. So there is a way to select from which type of re-referencing we have to do, but most commonly used is average re-referencing, where all the electrodes average is subtracted from each of the channel or electrode data. So now as I have mentioned previously, we have events. So we have read events file, but each event is associated with a code. So when I run this, we have events variable. Events variable has it's like an array, it's like a set of numbers storing what type of events are there. So there are, when you do unique, right, there are multiple events, repetitions of same events. For example, let's say stimulus onset is 1, then auditory stimulus onset is 2, then response onset is 3, and few more events are there, 4, 5 and 32. So they'll be repeating every trial. So when you put unique, it will just take one of the value instead of printing all repeated values. So we see here that these are the event codes used in this task. So what does the event 1 say? Event 1 says that the auditory stimulus appeared to the left. So if you have a continuous EEG data, wherever you see this one, that's the auditory left stimulus appearing. And 2 implies auditory stimulus appearing on the right. Visual stimulus appearing on the left is 3. Visual on the right is 4. Whenever the emoji comes, the smiley face, it's the 5. And whenever the subject responds, it's the 32. So basically what we do is, yesterday we have seen the trigger port, right? So to the trigger port, we write these values using the acquisition system, and it will send the trigger there. Okay, so these are the trigger values being used. Now we use these events to segment the continuous EEG data. So if I run this, we are just creating a dictionary for for later on plotting purpose or anything else. So right now it's not of important, we only need this. So, and now what it is doing is many epochs. This function takes in the raw data, uses the event names or event IDs, and takes 300 milliseconds before that event and 700 milliseconds after the event. So minus zero point 0.3 is going 300 milliseconds before the event of interest. So here our interest is all the events. So for each event we take 300 milliseconds before and 700 milliseconds after. Okay, and when I run this, it will just epoch the data right now. And now what we'll see is epochs.plot on the continuous EEG data, it plots these events. Okay, so if you run this epochs.plot, so you can see these lines, right? If I zoom in a bit, so this is the event ID and corresponding line. So there are multiple repetitions of this, right? So that is why you see many repetitions, and these are like there are many overlapping events. So since we are epoching with respect to every event, it looks like this. uh, 1 second. Okay,
How do you zoom in here?
just pinch using, I don't know if it is mouse, but if you have a trackpad you can just pinch and zoom, or you can just click on this and zoom. Yeah, you can just use zoom here. Click on the these three dots and then on your browser and zoom in. Okay, so we're just epoching, but now we want only epochs, like the events, that is 300 milliseconds before to 700 milliseconds after a given event of interest. Let's say my event of interest is auditory left. So whenever the auditory left stimulus appear, that is the event of interest, and you're going 300 milliseconds before and 700 milliseconds after, and like that you get for multiple events. And right now you don't have to worry, this is just the thresholding where whichever is a bad trial, what we are doing is if the amplitude of the given trial or channel is above 100 microvolts, then we are going to drop that channel, or yeah, or trial later, but it's mostly channels right now. So when you run this, it rejects few of the channels. Okay, so basically this shows from this electrode there was a bad epoch. Once you segment the EEG continuous EEG data from these channels, you got bad epochs, and based on that we drop 17 bad epochs. So here the epoch is nothing but from the event of interest 300 milliseconds before to 700 milliseconds after. So this shows here, you can see. So now it also like epochs after doing this, it also gives the epoch information. Okay, so these are the epoch codes, then what is the range of from each event, how much we are taking the signal, and sampling frequency as we have seen before, and these are all same as the before in data set information.
Hi Deepak.
Yeah.
There is this doubt by Arushi. She missed the part about re-referencing.
Okay.
So she,
We will come back to that. Right now, it's a part of pre-processing. You just remember that we have a data which is pre-processed. So yeah, as we go through the lecture, we will get there. So yeah. Yes, yes.
So we are basically rejecting the trial in any channel if it crosses 100 microvolt even once. So we are calling it a bad trial
Yes,
because the activity has gone beyond 100 microvolts.
correct.
Like, what is the rationale for that? Is it some
So
artifact of muscle movement or something?
yeah, it could be muscle movement artifact, or sometimes it happens because of the instrument noise itself. uh, like sometimes each channel, it reaches the amplitude reaches the cutoff of an amplifier. So those cases, like there are some hardware issues which can happen, very rarely. So then it can happen. And also we take a threshold based on number of channels also. Like at least if 15 channels are bad for an epoch, we just reject that epoch. That means that epoch itself is bad. If one channel is bad, we remove the channel and interpolate that channel. So right now I'm not dealing with that interpolation because this data has already been pre-processed. So just to show that like this is a kind of thresholding where you remove the epochs if they are bad. Otherwise it might affect your results. So yeah, sometimes you might not see a result also which you're expecting, or bad ERPs you see, when because this one outlier will drive the whole result. So therefore it's important to reject bad epochs. Okay, so now what we're doing is we are just averaging all the left auditory trials and left visual trials. So we have different epochs, right? Based on left auditory event we have some epochs, and based on left visual events
Just
we
one
have some.
observation. Deepak, I see that EEG07 sort of appears many times in the rejected epochs. Does that mean that the channel itself might be
Yeah,
bad?
yeah, it is bad, and it has more probability of giving bad data. So sometimes it happens, like as you use, no, sometimes the gel goes inside, accumulates, or uh, it can be of many reasons why an electrode can go bad. So many times you can see like same electrode going bad rather than different electrodes. So yeah, you're right. If it, yeah. So now what we are doing is all the epochs based on left auditory and left visual stimulus, we are averaging it. So this function so takes the epochs belonging to auditory left stimulus and then averaging it. Okay, same for the visual left. So we have averaged it. So L AUD and L vis has the average signals. Now we will see by plotting what we get. So we are averaging across the epochs, not across electrodes or any other dimension. So basically across trials, all the trials belonging to left auditory trials and left visual trials. So now it's just you give the data set and do dot plot, it will plot that. This is a part of MNE library, so you don't have to worry what is happening internally. We will see as we go on, but right now I don't want to give the MNE library details. So what you see here is uh, this is first one is the auditory ERPs from different electrodes. So you see a topo plot here. Topo plot is nothing but you plot electrodes on a two-dimensional head model. So this topo plot, and the color, corresponding color, is shown by this color on the line plot. So the y-axis is the voltage and x-axis is the time, and 0 is when the left auditory stimulus occur in the task. So right after the auditory stimulus occurrence, you see a nice ERP coming up, and since it's the left auditory trials, you will see higher ERP on the right electrodes. So if you're aware of contralateral representation of the world in the brain, that is left field of your real world is represented by your right hemisphere and vice versa. So you see that the red or pink colored electrodes have higher ERPs, and the other side has more negative or close to zero here. So from each of the channel we are seeing the trace of that ERP for left auditory trials. Similar, usually auditory responses show this, just one peak like this mostly, and then you see a flat or some end things like this. And now if you see visual ERPs on this figure, you see like 0 is the occurrence of that visual stimulus, and you see a positive going peak and then going to, it's called P to N. So it goes positive and then goes negative, or it depends on which electrode you're using. Since it's a left stimulus, you have this kind, and they have opposite directions because of the hemifield differences. So the green ones will be negative version of the purple or pink ones. So this you see is a visual evoked potential. This is an auditory evoked potential from different channels. uh, I hope this visualization is clear until this. If you have any doubts of how we are getting plots, please let me know.
Deepak, could you remind how N to P and P to N latencies work in left and right?
Okay, it's like, let's say you take this pink electrode, one of the electrode here. So P is positive going peak, and then N is going negative. So uh, P2 is at 200 milliseconds, you have a positive peak, and next immediate peak is the negative peak. So that is how you, that is why you call it as P2N, or if first going is negative and then goes positive, it is called N2P. So N occurs at 200, then P occurs. So this N to P, P to N are usually the visual evoked potentials. So whenever a stimulus comes up on a screen, you see a nice P to N or N to P depending on which side of the stimulus, or if it is central, you see most of the electrodes showing the N to P signal. Okay.
Deepak, why had you said that in visual you see it go down? Why does the P2N occur?
Oh, why does it occur? We really don't know the shape why it occurs, but it's mostly due to the visual cortical neurons responding to the stimulus. So whenever they get active, you get a, so these currents, the voltages here represent, like, if two neurons communicate, right? If you put an electrode there, if there is a excitatory response, you get a positive and then inhibitory. So whenever the stimulus appears and then goes off, there is a excitatory inhibitory response occurring. So that is why you get whenever a stimulus appears suddenly. So yeah.
Okay, cool.
Yeah. And now what else we can do with this ERPs? We want to understand, let's say this auditory potential, how across cortex all the electrodes, the cortex is responding to this auditory stimulus, because the electrodes which are close to temporal are more responsive to auditory stimulus. So what you see is more closer to temporal, you get a higher voltage values, and when you go to prefrontal or other areas, it goes less. So at a given point in time, how does the uh, brain activity look like? Brain activity here is the EEG activity. So what we are taking here is,
Deepak,
yeah.
it will be good if you can lower your camera angle, like so that we can see your hands. When you are explaining, you are using
Oh,
your
I
hands.
see. Okay, okay, sure. So is it fine now? Yeah.
Yes, yes, better.
So now what we want to do is, at a given point in time, how does the response of the brain look like for a given type of stimulus or for a given ERP component? So for example, here for left auditory stimulus, we are plotting a topo map using the time minus 200, uh, at centered at 200, minus 200, at 100, and at 400 milliseconds, and we are averaging the ERP in this 50 milliseconds. So for example, let's say this is 200, you draw a line here centered at this, you take a window of 50 milliseconds, and you have average activity in that 50 milliseconds window from each of the electrode, and that you do a topo plot. So then you get, like, initially there is no stimulus because it is minus 200, so you see a baseline kind of activity, not much difference. But as you go on, 100 milliseconds, 400 milliseconds, you see that the activity, and when the actual ERP is there, as you can see here, so initially at 0 or 200, then 400, So you see how the EEG response is in the cortex. So this shows at different times, at minus 200, at 100, and at 400 milliseconds from the stimulus onset. So this is another thing we see usually with ERPs, like which part of the cortex, or which set of electrodes are contributing to this particular ERP more at this particular time. Similar way, you can do a joint plot at different, like at this plot point, how does the cortic topography look like, and similarly at this point, how does the topography look like, and so on. So as you can see, it is more temporal here. Here it is temporal, but it is spread because there is interpolation and all we do. So, and also volume conduction happens, and whenever the potential is very high, the volume conduction will be even more. So that way you see more spread activity, but has actual thing, it weakens and it is more of a stimulus related one. You see more temporal electrodes being activated here. So I hope this topo plots are clear. This is another type of visualization that we do from ERPs. Is it clear? Okay, fine. Okay, so this is one level of averaging, that is across trials we do for different electrodes. But also if you have a region of interest, let's say this is our region of interest, I take this 4 or 5 electrodes in this, I hope you can see the cursor. So these 5 electrodes you take, this is the region of interest, and average across all those. That gives you a one single trace, giving you more, more reliable information about the event related potentials. So here what we are doing is we are defining like 4 electrodes for each side, left and right hemispheres. So and here left IX and right IX will pick the channels. So MNE.pick will pick the channels, and then you have a, like you create a dictionary to just use it for the plotting, etc., and combining the channels or averaging it. So what we do is we're combining channels using averaging in the ROI of interest, that is either left or right, and we actually loop it in the dictionary so that like one sentence, one line of code, you can plot both the things. And L audio is the auditory related epochs that we are using. So when I run this, you get 2 ROIs for each of the either left ROI and right ROI, and you see their ERPs here. So this is the time, this is y-axis across the region of interest, that is this 4 electrodes. And now what else you can do is you can compare 2 conditions also. For example, let's say I want to compare audio versus visual evoked potentials. What you do is you plot these things for both the electrodes averaged across a region of interest here. So this is how in general, like in neuroscience, we compare the ERPs. If you have 2 conditions, let's say one where you're paying attention and one where you're not paying attention. Yeah.
What is this region of interest? Is region of interest in time?
No. Region of interest
Or
is
in,
the set of electrodes that are of your interest.
oh, set of electrodes. So
Yeah.
yeah, somebody asked that what is ROI.
Yeah.
And another question was like what is topography?
Okay.
And what?
In geography, you might have heard this term, but we use this term in knowing how a map of electrodes and their responses look like. So topography is nothing but how a landscape look like, you want to know. So you have isoclines and all. So that is a geographical topo map, but this is a like neuroscience topo map, for example. So you can see here, it's nothing but a 2-dimensional plot with electrodes in their locations, corresponding locations of their electrode locations, and you see like iso lines here. So these are the lines which have the same activity, same amount of activity, same amplitude of ERP, So like that you draw. So this is the topograph or topo map that we usually call. Is it fine?
Yeah, and other questions was what like what are these averages of the above plots that when you plotted the topography plot, I guess. So
Okay.
yeah, these ones,
So
the three.
basically, you let's say I center around this line and take a 50 millisecond window here and plot each of the value on a 2D map and then interpolate it. Like this is a picture, right? This is a one dimensional view and this is a two-dimensional view. So at each point you get one value and that value you map
But
as a
that is
color.
averaged across trials,
Yes,
right?
averaged across trials. Yes, averaged across. This is auditory stimulus. So all the epochs where left auditory stimulus appeared, those trials you average and you get this, and then in a window of interest you plot this. This is in a window of interest. This one, whatever we are plotting. In the next one, we are seeing region of interest. That is in the electrode space. You are selecting, let's say, left electrodes. These are my occipital electrodes of interest. I'm taking them and plotting for left and right how they look like. So each of this line corresponds to, like first line corresponds to the left electrodes ERP, and next one corresponds to the right electrode ERP. Okay, fine. And after this, we are seeing if we have two conditions. Right now, we are only comparing auditory and visual. So auditory ERP versus visual ERP, and we just plot their ERPs and compare. Also, another way to look at it, this is you have an attention task where people pay attention on few trials, and few trials they are not able to pay attention, and how their ERPs looks like. You can do those things also if you have an attention task. This is just a sample task for playing with the data. So you can also plot like this is a population mean, right? So you can also plot to know how confident you are, the variance or the standard error. So we will deal with these terms. Right now you remember it as how variable is the data in that window. So the size of this bounded color represents the variance in the data, so that you know which part you can be more confident of. If the variance is more, that means uh, that amplitude is not very reliable and it has a longer distribution or something of that sort.
Deepak, the plot with the mean above this, didn't we establish that visual component goes from P to N? In this plot, it's the other way around, or am I getting it wrong?
Okay, it depends on which electrodes they are using. So if you take 10, 11, 12, 13, 14, so if you see where they are,
Mm-hmm.
1 second, where are these electrodes? Mm, where do you see 10, 11, 12? This ones, right?
Frontal.
Oh,
Yeah,
these are
yeah, okay.
frontal electrodes, acha. So that is why
Mhm.
the ERP is not very clear also. If you take these electrodes, no,
Yeah, yeah.
you see more clear. But usually there is what is this called? A discharge happening from the visual cortex because of eye movements and visual effects also.
Mhm.
So
Got it.
even the activity here can be captured using this electrode. So because of volume conduction, and we'll look into how to minimize it in the later classes, and
Mhm.
we'll see a small intro to volume conduction here in this lecture also. I hope we
Okay,
have.
so you're saying if we had chosen electrodes from 44 to 49,
Yeah,
we would see.
correct. If we chose these
Okay.
yes, correct. Yeah, yeah. You can try
Why is that not
also.
audible?
We can select also. Can you please tell the electrode name numbers?
It's 44, 45, 46, 47, 48, 49, 50.
45 to 50 I can write, right?
Yeah, yeah.
Okay, let's see what. Here you can see, right?
Clips, yeah.
Yeah. So yeah, the more you average, the actual thing you get. So overall amplitudes might differ because as you increase the number of averages, the amplitude generally decrease. But you can plot a, this is just a mean, but you want to know how confident are you are with the thing, so you can plot an error bar also around it. So we can look into how to plot this later, but right now it's only about visualizing this and loading the data and seeing what ERPs, what all things can we do with ERPs. And so I hope this notebook is clear. You can execute by yourself once and get familiar with it. And if you have time, you can look into what each function does also. We'll go into each as we go through the lectures. So I've shared this notebook. You can look into this. Okay. So sorry. Yeah. What is happening here? Okay, fine. So this is a task that we have looked into. One second.
Deepak,
Yeah, yeah. I think one second.
So I tried using just the right hemisphere electrodes 49 to 52.
Okay.
That shows a better graph of the
Oh, nice.
just the.
Yeah, yeah. Great. So I think I have to do a window. Yeah. Okay. My screen is visible, right?
yes.
Okay, great. So, okay. Oh, I don't think we have much time, but I'll take another 10. No, we have time, right? It's until 8:30. Yeah. Okay. So now we will look into some interesting aspects of pre-processing to get more clear and what's time frequency representation. Uh, yeah. So any signal can be represented as a mixture of sinusoids. What I mean by that is you'll get to know now, right now. So here you see, like let's take an example of a music signal. Like music is nothing but compression and rate fractions in the air. So I'm plotting the, on the y-axis I'm plotting the pressure in the air and as time progresses. Let's say you have different notes, D 294 hertz, A 440 hertz, F 349 hertz, and these are different notes in that music signal. And when I'm playing all the chords or notes simultaneously, what you observe, uh, one second, sorry, this doesn't play. Okay, yeah. So this all signals add up and you get a signal like this. So this is what the air compressions and rate fractions look like when you combine these three signals and you get this complex waveform. So just by looking at it you can't tell which which notes are there and all, but when you actually combine you see. So what is this saying is any complex signal of this form can be represented using sum of sinusoids. So what I mean by sinusoids is repeated periodic oscillations of a value, or for example here the pressure, the pressure, whenever you start playing, the pressure starts like this and you see a sinusoid like this. So anything going like this in the sinusoid fashion we call as sinusoids. And so if you're not clear with what is sinusoid, I'll show you. So here you see a GIF here. This is like a simple harmonic motion or a rotatory motion, and whenever you plot the value of this dot moving, how do I stop this? I can't stop this, but anyway, So you see the amplitude value. So whenever the arrow is here, you get a this value, and whenever it is close to this, you get a smaller value. And you plot on a two-dimensional plane, you get this
We can't see
sinusoid.
your cursor,
Oh, is it?
by the way.
Now can you see?
Yes, yes.
Okay, fine. So this harmonic motion, when you plot on a two-dimensional plane, you see a cycles going like this, and these are called sinusoids. They can be mathematically represented as a sine omega t. Omega represents how fast is this rotating on the circle. So if it is faster, you see within smaller time you see more cycles going on. So this is a general expression of a sinusoid, and basically the rate of represented by omega also represents the frequency. So how many cycles per second you have. So omega can be represented as radians per second or cycles per second when you multiply by a 2 pi conversion factor. So this is what the f represents here: 440 hertz, 294 hertz, 523 hertz, and when you combine all this, you get this type of complex signal. So now you can imagine there is an EEG signal like this. You can use some mathematical techniques to split this signal into its sinusoid components. So basic idea or takeaway message from here is any signal can be decomposed into sinusoids, and any signal is a mixture of sinusoids. So uh, and we'll see time frequency representation soon. So I hope this part is clear. If you have any doubts, please ask it here itself, because we're going to use this extensively in next classes also. And it is also important to know this for filtering and other pre-processing steps. Okay, hope it is clear. So this type of decomposition is also done by a mathematical technique called Fourier transform, where you give a signal and you split into its component sinusoids or monotonic oscillations. So now how do we visualize? So for now we are not dealing with what is this mathematical technique, but you just remember it's called a Fourier transform. And whenever there is a signal, let's say this is a 2 hertz signal. What I mean by 2 hertz, you have 2 cycles per second. So you have two troughs or two peaks in 1 second. So 2 cycles basically. You see, if you start here, this is 1 cycle and this is another cycle. So in a time of 1 second, you have 2 cycles, and therefore it's called 2 hertz. And in this kind of a signal, when you do a Fourier transform, so this is a Fourier transform, it's like a measurement, so it's not exactly, it's an estimation of it. So what do you get is, you get a small peak like this. When you transform it, this axis becomes frequency axis. Unlike this is time axis, this is a frequency axis. You see a peak at that particular hertz, and that is the 2 hertz thing when you do a Fourier transform on this sinusoid. Similarly, if you take 3
So if it is a pure harmonic, basically pure 2 hertz signal,
Yes.
then we should get a like
Yes.
one single peak, right? This
Yeah,
must be on some noisy data.
no. So that okay. This is done in a computer. So what happens is, so this is not a nice continuous signal that you're seeing here. It is actually a digitized signal. So the Fourier transform that you're doing is a discrete Fourier transform, or discrete Fourier transform. So discrete Fourier transform is not exactly a Fourier transform. So it's a algorithm that is an approximation of a Fourier transform. So because of that physical implementation, we don't get a nice one line here, which is like, you can never see a one line here because that is impractical. So in practice, whenever you're doing a Fourier transform, you generally see this type of a peak here if you have a monotonic signal.
And there is some other doubt in the chat.
Okay.
Mhm. What is the difference between time and frequency domain? And why to move to frequency when we were in working with the time domain? What was the problem with time domain?
Okay. So There is essentially both are same. You know, a signal, whether you represent in frequency or time, it's the same signal you're representing. So it's not giving any information. You can think of it as a visualization tool. So right now when I show you this, that each of this signal you add up, sorry, yeah, uh, one second,
That pressure wave,
yeah, why is this
where is it
not-
gone?
one second.
Yeah, but probably you can recall that there was a pressure wave.
Yeah, you see a complex
It
wave
was
when you
not-
add this up. So in that way, when I show you, if I don't show you this, if I don't show this lower three waves, will you be able to tell which components or which frequencies or which sinusoids does that signal contain? So like, you can't tell if I hadn't shown you this. So that way, like when you do a Fourier transform, when you convert a time domain signal into a Fourier domain, from time domain to Fourier domain, you see a frequency domain, you see the peaks at this. So for example, here, like if I show you this, you can count. If I show you only one monotonic oscillations, you can count and tell, but that is a lot of effort, right? You can count even in that complex signal, you can count and take an LCM, or you can do that, but to the naked eye you can't do it. So for that we need a tool to make it same signal transform into its Fourier or frequency domain. So when you apply a Fourier transform, you convert time domain to frequency domain, and you see at 2 hertz you see a peak. So if I show you this wave, you say that this signal has a 2 hertz. One second. Yeah. Yeah. Is it fine? Yeah. So similarly, you take 3 hertz, you see a peak at 3 hertz when you do a Fourier transform. And when you take a sum of these two signals, as I show you here, here, like if I show you this signal, we can't tell which frequency it has. But if I do a Fourier transform, you get exactly two peaks here at 2 hertz and 3 hertz. So this is a this is a frequency axis. So then you get two peaks here. So is it clear, like why to see a signal, same signal in two domains, time and frequency?
So the doubt is that why are we moving to this? That is not still clear. Like, what,
Okay. So yesterday we were discussing why we are moving to frequency domain.
yes, yes.
Okay. So so we do a lot of pre-processing steps where it has band pass filter and all. So to make it clear what is happening, and also like we have discussed about neural oscillations, right? So neural oscillations are nothing but these kind of oscillations. You want to see whether it's theta or which band is prominent. For example, in a memory task, usually it is found that theta band oscillations or uh, are like, you see a theta oscillation going on when you're registering something in your memory. So whenever you do a Fourier transform and plot, you see that theta band, you see a high value in that theta band thing. So in order to know which kind of oscillations is there in a given signal, we do that. So yeah, these are the two, right now these are the two things for which we are like right now going to Fourier frequency domain.
So my add-on to that answer will be like um, this regarding neuro oscillations. These different frequencies are hypothesized to be for different to play different purpose in the brain activity.
Yeah.
Especially studied in sleep, but also in awake period they play different roles, and they can be modulated by some stimulus, external stimulus,
Correct.
or perturbations. Other perturbations are not stimulus. So that's why, and we can see a signal, and then if you present something, then you can see that which kind of frequencies are going up or which kind of frequencies going down. Then
Yeah.
you get a better idea instead of just analyzing the time and values.
Yeah, correct.
So seeing that peak after stimulus, but seeing that peak in what frequency. So if you do a Fourier transform, then what particular frequency is changing, then you can say much more about the uh,
Yeah,
stimulus.
or yeah, or the
And
state of brain. Also, here for just for making it simple, we are only using one like amplitude of one oscillation. But if the same oscillation is of higher amplitude in some time and in other times they are low, you can also plot a 2D Fourier transform kind of thing. We'll see that later in the future classes, but there you can see at which time this particular oscillation is more prominent, at which time this is not very prominent. So that way you can also compare, uh, like you can also infer about what state of brain it is. For example, alpha waves are known to be a sign of, when you just relax or close your eyes. So you can also tell when a person is sleeping just by looking at their alpha profile in the time and frequency.
so there are other doubts which say that uh, why are you using Fourier and frequency terms interchangeably? So I don't get it fully. If you can unmute,
Okay.
probably it will be great. Like we can
Yeah,
discuss
please.
better. But I
So,
don't
yeah,
get like.
yeah,
Yeah,
hi.
yeah.
So, while describing this frequency domain thing, I mean, many a times Deepak used
Yeah.
Fourier and frequency interchangeably. So, does this mean that the Fourier transformation can only be carried out in the frequency domain? Because, as Athar wrote in the chat, that we conduct the Fourier transformation just to, I mean, split a signal as a summation of the many sinusoidals.
Correct.
So yeah, so now the thing is there any relation in between the Fourier and the frequency thing? Number one, and in the frequency domain, moreover, I mean a single peak is being shown.
Yes.
So I mean, whereas in the time domain we have many peaks and many valleys. So I mean, what's the meaning of this
Okay,
single peak
okay.
in frequency domain?
Yeah, yeah. So, coming to the first question, I've been using Fourier and frequency interchangeably because that is the like uh, there are wavelet based techniques also, but even then, the base technique to split a signal into its frequency components is using Fourier transform. So, like in lot of like everywhere, Fourier and frequency became interchangeable over time. Uh, but yeah, it's always better to say only frequency. So Fourier transform is a technique and frequency is a quantity. So yeah, that is first one. And second one, you see many peaks here but not here. So okay, basically the same signal but information that you're seeing is different. So in the time domain, you see how so frequency is how fast the signal is fluctuating in the time domain. So the more fluctuations you have, this peak will go to higher frequencies. So since it is only one frequency, you get only one peak here. But if you have if you take a sum of this, you see two peaks, right? And similarly, if you have a very complex signal, it's a sum of many sinusoids. There you don't see a like clear peaks like this, rather you see a continuous waveform like this. And yeah, I hope that makes it clear. So it is
Yeah.
nothing
So
but
is
how how the signal is fluctuating in the time domain is shown using a different axis or using this Fourier transform.
So, is there any meaning of the relative position of these peaks? Like, say, in the frequency domain, quite possible, I may get these peaks quite closer. I mean, and sometimes they're quite
Yes,
far apart.
yes, yes.
So, what could be the meaning of that, or the interpretation of that, or is there any
So
meaning? I mean, of the significance of the distance in between these two peaks here?
yeah, yeah. If this signal, if I do a sum of 2 hertz and 2.5 hertz, you get a close by peaks, or basically overlapping peaks, and you get a broader peak here when you sum a signal with 2 and 2.5 hertz. So so basically this 2 hertz is this value, the x-axis value here, and 3 hertz is the x-axis value here. So if they are close by, you get close by peaks. If they are far apart, you get farther away peaks.
Oh,
Deepak, I
I see.
think one more thing you can touch on is that in the time domain, we know what is the meaning of magnitude or the height of a peak. It's just in this case it can be neural activity. The higher peak means there's more activity. In the frequency domain, currently here there are two peaks, but they are of the same height. What
Yeah.
would it mean if different peaks have different heights in the
Okay,
frequency domain?
so right now for simplicity, I'm showing same amplitude, like amplitude one sinusoids being added, but if I take a some two microvolts oscillation like this and 1 microvolt oscillation like this, you will definitely see this peak being higher than the 2 hertz peak when I add the signal this way. So it shows the power of the oscillation, like uh, you have 3 hertz with higher amplitude, that signal will have higher power compared to the uh, if you take 1 volt 2 hertz oscillation. So this amplitude of this shows the power of particular oscillation. So if you increase the amplitude of this sinusoid, that particular peak's amplitude also increases.
So I have one more question here, that I mean related question, that why don't we have this peak in the beginning itself, and why do we get in the middle, or sometimes towards the extreme?
Okay, so if you have a 0 hertz DC shift, like this signal is at zero, centered at zero, right? So if I add a let's say three volts to this signal, it goes somewhere up. So 3 volts DC, that is a constant signal, is nothing but a 0 hertz signal. So if there is a DC shift here, if I add some constant value to each of this, you will also see a peak at zero. Let's say this is zero, you will see a peak at zero and also peak at one, peak at 2 hertz or whatever frequency that you're looking at. So if I add a constant value to this signal, you will also see a peak at 0 hertz. But right now
Thank
the DC is zero.
I sense a fundamental doubt in Mr. R.K. Goyal's questions, that the left side plots are plotted on time axis and the right hand plots are plotted on frequency axis.
Correct.
So that's where the difference comes in. Like, it's not, so you should
Yeah.
not read in that the frequencies, the peak is coming later or
Correct.
earlier. It's in frequency domain, so any peak can appear. Like, if something appears at 0.5, then it will be sine of 0.5 t.
Correct.
That is just
Pi into
a
0.5.
signal with a lower frequency, from time 0 to whatever time you have measured.
Yeah.
So
So
that's the fundamental
yeah.
question that I think is generating his doubts.
Correct. So these axes are entirely different. This is time. It shows like as time progresses how the voltage of a signal goes or how the music's air compression goes. But here it shows a different axis. It's a frequency, like what kind of oscillation is present in a given signal, and like yeah, based on like what Atharva told, it's the fundamental differences in this axis itself. Like you should not compare like why this is like this but not this one. So these two are uh, information in the same signal, but two objects in that or two attributes of the signal, of the same signal. So two different attributes of the same signal. One is frequency, one is time.
So to conclude with what Deepak has told, a signal is a combination of sine wave with different frequency. So when you are understanding the signal in the frequency domain, you can understand the frequency components which is present in the signal. So with respect to EEG, if we talk, we can confirm that signal is in the range of 0.5 to 40 hertz or so, which is based on our study. And also we can understand whether any noise is present. So if there is a power line noise, we can find a peak in the 50 hertz so that we can use appropriate filters to remove that. Am I right?
Right, right. So
Yeah.
thank you for the input. Yeah, whatever. Yeah. So can I know the name, please? I couldn't see the teams.
Yeah. Divya.
Oh, Divya. Thanks, Divya. So, whatever she told is right. So, if in an EEG signal, if you have a line noise, you will see a peak at 50 hertz. I will also show like how a EEG spectrum looks like as I go on, and it will be more clear then. Yeah. So now we will look at some complex signal. Let's say you have a signal like this with this many fluctuations, and it has multiple frequencies. And when you do a Fourier transform, you get these components. These are the oscillations, different oscillations present. And let's say, like as Divya told, there is a line noise. Let's say this is 50 hertz. In this signal, if there is a line noise, you'll see a peak at 50 hertz. Right now, in the processing of the signal for later analysis, you don't care about this noise. It's a noise, you don't want this. So what you do is you do something called filtering. So that filtering is nothing but it's called removing certain frequencies or a particular frequency. So if I want to remove a line noise, what do I do is, in the frequency, like I convert into frequency domain, I create a filter which cuts down any part beyond this line. So that is called like low pass filtering. You're passing lower frequencies but not frequencies above this. So if I draw a square box like this, you're going to pass this but not these frequencies, and when you see that filtered signal like with only these peaks, you get a smoother version. So this is a signal with noise, like highly fluctuating noise, quickly fluctuating noise, which is shown by this peak. But if I remove that peak using some filtering, you see a smoother version of the same signal, right? So this is what we call as filtering in the pre-processing steps. So you can also put a band pass filter, like you are not interested in very low frequencies or very high frequencies. Then let's take a filter like this, which you draw a square like this and multiply that, and you get only these three peaks, and that becomes a band pass signal of this particular signal. So depending on what frequencies, whether you're passing lower frequencies or higher frequencies or a band of frequencies, you call it as low pass filter if you're passing low frequencies, high pass filter if you're passing high frequencies but not low frequencies, and band pass if you're passing certain bands, and you can define those values as you require. I hope this picture is more clear of what it means, the frequency domain.
Yes, it's very clear.
Yeah, cool. So this is the filtering part that we generally use in pre-processing steps yesterday. Okay, I hope this is clear. If anyone has doubt or any queries until this, you can ask, please. Okay, so this is just an intuitive view of how to see time and frequency representation of a signal. So this is just a picture view, but if you take a real EEG signal, it's not this simple. The spectrum also looks very complex, and in a bit I'll show that too. Also, we have seen something in the pre-processing steps here, as you see here. Like here, you see something we call sampling, downsampling, and this box, ADC box. So generally the physical signals are continuous signals, but you send it to the computer or any storage device using samples of it. So we don't send all like full continuous signal, we can't send to a computer. So we need to do sampling. So let's discuss a bit about sampling. So just to say that there is a particular rate at which you can sample so that you don't lose any information. So that is called Nyquist rate. So it will be more clear as I discuss. So let's say you have a signal like this. This is some signal you want to sample and send it to, some physical signal you want to sample and send it to your computer. So you can sample in many ways. You can take one sample here, one sample here, one sample here, in this way, and send it to the computer system. But let's say the receiver is someone else who doesn't know how the actual signal looks like. So now he tries to join these lines. So now if you try to join these lines, you will see a different signal. So like you can sample like this, but here you're not preserving anything and the information is entirely different. You can also do similarly of this sort, and you can see that you can still construct approximately same signal if you sample this way close together. And if you sample even closer, if you sample each and every point on each of this half upgoing part and downgoing part, you see that you can even perfectly reconstruct the signal. So the rate at which you can sample so that you don't lose the information in the signal is called Nyquist rate, and it is generally called it is 2 times the Nyquist rate is nothing but 2 times the highest frequency present in a given signal. Let's say this signal is of 2 hertz, so you can sample only at 4 hertz to reconstruct the entire signal. So 2 times 2 hertz. Like that, if you have a complex signal, it is like uh, let's say you have a complex band limited signal. Band limited, I mean it has only few frequencies in that signal. So let's say that maximum frequency is 100 hertz in that EEG signal, whatever we have dealt with, or 150 hertz. The Nyquist rate of that will be like 2 times the 2 times the bandwidth of the signal. So this is the Nyquist rate. Uh, this is how you sample. There is a minimum rate at which you can sample, or you can sample above that. You can also sample even more than this, but you can't sample less than this because you will lose the information if you sample less rate than 2 times this signal frequency. So this is called
But
sampling. Yeah.
sorry, but so this has a continuous amplitude over time.
Yes.
What if it changes in very short times? How do you decide the Enquist rate then?
Yeah. How do we decide the Nyquist rate is knowing the maximum bandwidth of a signal. So for example, I'll give an example with two scenarios. One is you're sending a communication signal, that is you're sending a message or you're talking on a phone. Those signals are of very high frequency, let's say few kilohertz, okay, 1 kilohertz let's say. And you have EEG signals. EEG signals are usually like are the neural phenomena of interest are usually below 80 hertz let's say. And now you know the limit of the signal. So EEG signals limit is 80 hertz, so the bandwidth of the signal is 80 hertz, and whereas communication system signal bandwidth is 1 kilohertz. So now the Nyquist rate of these two is the communication signal, which is 1 kilohertz or 1000 samples per second, is 2 into 2 you have to do, that is 2 kilohertz. So the communication signal you can sample at least 2 kilohertz or more, only then you can reconstruct the whole signal. But if you're taking EEG signal which is only 80 hertz, so you should at least sample at 160 hertz or more, you can't sample less. So you have a maximum frequency that a signal can have depending on its physical or the structure of the signal. So based on that you decide the bandwidth of the signal, and based on the bandwidth you decide the Nyquist rate. So if I say bandwidth of a particular signal is 5 hertz, the Nyquist rate is 10 hertz, and that means you can sample at 10 or more than 10 hertz. Only then you can construct the whole signal, otherwise you will lose the information. Is it clear, Arushi?
I'm still confused about whether if the same sample that you're looking at has two different, like you said, one is 1 kilohertz, the other is 80.
Yeah.
What if this happens in the same sample, or does it not happen? It would,
Okay,
right?
so if it is less, right? So let's say 1 kilohertz signal, it also has 80 hertz signal, right, which is lower. So you're sampling at very high rate, that is like if you say that 1 kilohertz signal has 80 hertz signal, 10 hertz signal until 1 kilohertz. So if you sample more than 1 kilohertz, you're also above Nyquist rate for the signals which are of low frequency. So in that case you don't lose, like that is why while sampling bandwidth of a signal is more important than what frequency components are present. So bandwidth is the maximum frequency present in that signal.
Okay.
Yeah. Yeah. If you have any doubts, we can discuss offline. Yeah.
Right. It was just one kind of a very small follow-up question. Talking on phone, let's say, they need to sample at least at 2000 samples per
Correct.
hertz,
Yeah.
but in the actual signal might have many, many more samples, maybe 5000 samples.
Yeah, yeah.
And so
You can have more.
then in improving the sort of increasing the sampling frequency further, will that lead to a cleaner, more audible signal, or is it just the same?
Yeah, yeah. So if you sample higher than Nyquist rate, right, you get even more, more resolution of reconstruction in the signal. So the more samples you have, the more perfect the signal reconstruction is. So it's like you send a discrete samples to other receiver, and receiver you reconstruct it. So if you send more samples, it is more clear. But the cost there is the the memory costs, or the overhead cost that you have is the memory each sample requires. So if say if you're sending 2000 samples and you can still reconstruct signal, I can send 5000 samples and still reconstruct perfect signal, but the cost there I'm losing is the the memory or the resources required to transmit those 5000 samples versus 2000 samples. So yeah, you get an even better signal if you send more samples. So yeah, so now like this leads to, like, if I downsample, let's say you have this signal at this sampling rate you have, but now because of storage limitations I want to downsample it. But let's say it is 1 kilohertz, and I want to sampling rate is 2 kilohertz. At 2 kilohertz I sent, but now I want to downsample it. So can I directly downsample it? If I downsample, I will lose the information if I go below Nyquist rate. So what in that case, what you do is you band limit the signal. That is, you create a low pass filter. You remove certain frequencies beyond that frequency, and then okay, I think it might not be very clear here. So I can use this figure. So what I do is, let's say I want to downsample below this much sample rate. I don't then what you do is you low pass the signal so that you have removed higher frequencies, and now it is you can comfortably downsample and still reconstruct the signal if your interest is of not the higher frequency signals. So that is one of the step that we do before downsampling. So if you see here, yesterday as we discussed this downsample the data. So while downsampling, you must keep in mind that the downsampling should not cause a problem while constructing the signal back. So further you need to band limit using a low pass filter before, and then do the downsampling. So it's also called anti-aliasing filter, or if you don't do that, it will create an aliasing effect. Is it clear? Vinod? Okay, we can discuss. Okay.
Hi Deepak.
Yeah.
You didn't explain aliasing, I guess, and you've used it.
Yeah.
So do you want to did you miss explaining, or do you plan to explain
I
later?
can
Just
explain later. Right now, I don't think we have time, but right now our interest is this one: what is sampling. We'll discuss the downsampling in next classes. But before that, I want to show how a frequency domain of a typical EEG looks like. So it's the frequency domain or Fourier transform of an EEG signal is also called a spectrum. So here on x-axis, let's say I have an EEG signal, and then I do a Fourier transform on it, I get something like this. So the x-axis is the frequency, y-axis is the amplitude. Although we are doing some scaling transformation using decibels, so you don't have to worry about that. For now, you remember the y-axis is the power, and x-axis is the frequency. So a typical EEG spectrum looks like this. So and usually in most of the cases we see a peak in alpha, usually, like, because there are periods of rest where while subject is doing a task, you get a, there are many components which contribute this 10 hertz frequency. So that's why you see a peak here. So basically it tells wherever that frequency is present, you get a peak here. So let's say they are doing some memory task, you see a theta peak like the red one here. Then you can define that during this, like, you can do in certain time periods also. We'll discuss about that later, like what is the relation between time and frequency uh, in more detail. But yeah, if in your task they are trying to remember, you see a peak in theta, because theta is known in memory consolidation or registering certain memories. So this is how a typical one looks, and you might have a doubt that why is it going down as the frequency increases. There is a noise called pink noise, so that is nothing but as the frequency increases, the you know the amplitude or the power of that particular frequency drops as it increases. So this is all, this type of noise is present in all biological signals. We will also discuss why it is present if we have some time, but I don't think we have much time. If you give me 5 minutes I can explain, but yeah. So any doubts in like typical EEG spectrum, or why the characteristics looks like this?
Is it not that the brain activity is in the low frequency range, and this particular graph, like higher frequency components are less? Is it not like that?
Yes, exactly. It is just because of that. So it's like the number of components contributing to this are actually it's the opposite way, but we can discuss that in the next class. So it's like the more numbers you average, the average value reduces, kind of thing. So
Do those blips have any significance in the
this one's,
blue? Yeah.
yeah, they do have. So we actually, to reduce this pink noise, the decaying noise, what we do is we do some kind of normalization. So we take a broad, like, average of this whole spectrum and divide by that, or here if you're doing log scale you subtract it, so that you get a, like, you don't lose this, like that, basically you are eliminating this kind of noise. So or you can actually fit an exponential and do a reverse of that, so that it becomes more flatter, and you don't miss on, just because of noise you don't miss the important blips here. So although you see small, when you do a normalization you see like more, everything becomes flatter and you see a peak like this. Yeah. So these are peaks in beta oscillations. Beta oscillations are also known in some feedback processes from while making decisions, or in attention phenomenon. So this is just a typical spectrum. So if you know the task, I can tell you what significance this have, but yeah, this is just to depict what is it. Yeah. So I thought to talk about re-referencing also, but yeah, I can talk about 5 more minutes and then we'll close. So why we do re-referencing, as we have seen in previous notebook. So let's say you have an electrode. So it's not the absolute potential that you're measuring using these electrodes. Any potential that you measure is with reference to some other position, or some other electrode. So that is nothing but the reference electrode, or reference in that, if you have a EEG system, reference or ground of that EEG system. So ideally we assume that the potential here to be 0, and therefore whatever voltage we get is the voltage, EEG voltage. But in real cases this is not exactly 0. There is always some noise on the ground electrode also. Or so what happens is that noise to an amplifier, if you give that signal to an amplifier, the difference of this non absolute 0 and a voltage, you get some noise into the EEG signals. So, and also this electrode is usually somewhere else, it's not on the scalp. But in case you want to remove a common mode signal or common mode noise across same space, what you do is you take a reference on the scalp. Let's usually central electrode or temporal electrodes are taken because they usually don't capture very significant activity from the brain, but they're still close to the electrodes. So you take this as the reference, either this or average of these two, because you don't want to bias your data, and subtract that signal from every other electrode. Or other way to do is you take every electrode average and then subtract that from the signal, all the electrodes, not just few electrodes. That is called average re-referencing. So these are different types of re-referencing. We are using this because the ground is not always ideal. In real scenarios, ground has a noise, and to eliminate that noise, we need to do re-referencing.
So in this diagram, CZ and TP10 are the two electrodes that you subtract every electrode signal from.
Oh, either of them. I can use, this is called there are different types of re-referencing. One is monopolar re-referencing, that is you just select one electrode and that activity you subtract. Or you take average of all the electrodes, that is called average re-referencing. There is also bipolar referencing, that is average of these two electrodes are taken and subtracted, or usually two temporal electrodes are taken. So the TP10,
And it's established that these electrodes in every human will not give as good a signal. I mean, they will have no signal.
yeah, they will have some signal, but it is very far, so they're not mostly useful. And even this is center, so this usually have like the cancelling effect over the dipoles and all. So that is why there are standard referencing electrodes. This is center referencing or CZ referencing, or you use temporal electrodes, which are just above the ears, so they also don't capture much of the, and you take an average of this because you don't want to bias one hemisphere over other. So when you're taking temporal, it is the bipolar referencing. When you take CZ, it is unipolar referencing or monopolar referencing. And the other referencing is you take all electrodes average when you are using all the electrodes and subtract that whole average signal from each of the electrode. So these are useful to avoid noise from ground electrode or reference on the EEG system, which is always having some kind of noise. You can see one example here. This is a center re-referencing done between there is some frequent stimuli versus rare stimuli. The blue represents the rare stimuli, the black represents the frequent stimuli, and you want to see the difference between them, difference between the two events in a task. So when you do a central reference, you see this red has a difference wave, and when you do average re-reference, you see this has a difference wave. And when you do a mastoid, that is the temporal electrode average referencing, you see this kind of ERPs. So our interest is difference wave. So usually frequent versus rare stimuli, there will be a positive difference in the ERPs. So based on different re-referencing, you get different different answers. So any reference is not a standard. It depends on the use case and how many electrodes you are using in a given experiment and many other factors. So in this particular case, I just want to highlight that ground truth, we know that difference wave for rare versus frequent stimuli should be high when the stimulus onset happens. So this blue one represents the stimulus onset, and among these three, you see the B1 has the highest difference. So in this case, when they are using all the electrodes for the task and this kind of a oddball task, the best reference is the average reference. So based on the experiment and type of measurements you are doing, the re-referencing also changes, and 99% of the cases it is all average if you're using all the electrodes. In other cases, you have to go by your intuition of what makes more sense. So this is about re-referencing, and why do we need re-referencing, and what type of re-referencing are there, and which one to use when. So this slide summarizes it. So
What do you mean by mastoids? Which are those electrodes?
those are the temporal electrodes, as you can see here, TP10, and symmetrically the other side. These are just above the ears. So those locations are called mastoid. So the electrodes above that are called mastoids or mastoid electrodes. So those electrodes near the ears, don't just above the ears or behind the ears, are used for re-referencing. Yeah.
Hello.
Yes.
So, which of these referencing is represents the truest nature of signal?
Truest nature of the signal. So
I mean,
ideally,
like one of these, the last one, last referencing is the shows the major difference between one and two, right?
yeah, yeah,
The other two don't like. They also show some increase, but not to that extent.
correct.
So, like, what represents the actual thing that is happening in the brain?
Acha. So this difference comes up, but you also see other things happening here, right? So, and you, this is a, like I didn't mention it before. This is an auditory oddball task. So where they see frequent versus rare, and whenever rare stimulus occur, they identify it. So, like usually when you're using an auditory task, mastoids are not generally used as reference, because mastoids might capture auditory, you know, stimuli, auditory, effects more, the temporal electrodes. So you generally choose based on like what kind of task you're doing. If you're doing a visual task, then mastoids can make more sense, or average also makes more sense. So yeah, you choose based on what type of task it is. And although the difference is more, just because you're seeing the result doesn't mean this re-referencing is correct. It also depends on like what are the standards established based on the task. And
Okay, I see. So you choose the electrode which you don't believe that it will show the signal. Like for a
correct.
visual task, the auditory stimuli will not show. Auditory electrode will not
Yeah,
show the signal.
correct.
Should not show the
Should
signal.
not. So yeah,
And if that as a reference, that will be a better.
yeah, yeah, correct.
Okay.
But for many of the devices, there is a predefined reference, no? Or can we choose it according to our?
Okay, so this is called re-referencing because it is done like while collecting data. There is a referencing, like there are electrodes which are called common mode signal driven right leg electrode and all. So those are like online referencing, but post hoc when you're analyzing, you also do re-referencing to even eliminate any kind of noise that could be present, which is very different. So this is the common noise across different electrodes. So based on position of electrode, each electrode might have different noise levels and all. So so if you take this kind of referencing, you eliminate those kind of common noise across the electrodes. So that is why it is called re-referencing. But online also, when you're collecting the data, there is other type of referencing done, and this is another further step to even reduce any other noise effects. So
Okay, so
yeah,
this is after data acquisition.
yeah,
Got
this
it.
is called post hoc referencing or re-referencing.
Okay.
Yeah. So I think that's it for today. I've taken a lot of time, but yeah. So
Thanks, Deepak, for explaining, going through the notebook, and thank you, everyone, for